had one that I think they added to it. It was one we used in the past.
Good afternoon, everybody. It is so great to see everyone here from so many different departments across the college and graduate school. Thank you for coming. It's my great pleasure to introduce this year's Hubert McNeill Poteat Lecturer, Dr. Christa Colyer. The Poteat Lecture Series celebrates the research, scholarly, and creative achievements of faculty, and I wager that many of you in this room have witnessed firsthand just how Christa's work exemplifies that spirit and standards. Christa is a professor of chemistry and the James and Courtenay Harton Faculty Fellow for Chemical Industry. That was a mouthful. All right. Since joining our faculty in 1997, she's built a research program of remarkable scope and real-world impact. Focuses on incorporating carbon dot methodologies for drug testing and other practical applications. And I think one of your students came to the CBOV, gosh, it was only last week, and she had a dental application for this work. Am I right?
Yeah.
It was really cool. I'm so impressed. All right. Very, very cool. So she has served as principal or co-principal investigator on $3.2 million in external grants, published 75 peer-reviewed articles, and delivered 51 invited research presentations. 52 after today. We'll count this one. What makes Christa's scholarship truly distinctive, though, is that it has never happened apart from students. She has supervised 117 undergraduate research projects and mentored 18 graduate students and 3 postdoctoral fellows, turning her laboratory into a place where the next generation of scientists learns not just technique, but what it means to pursue science in the pursuit of others, in the fullest of the pro humanitate spirit. Her colleagues and students have recognized this again and again. She has received Wake Forest Reed Doyle Prize for Excellence in Teaching, the Teaching Innovation Award, the Award for Excellence in Undergraduate Advising, and just this year, the College Board of Visitors honored her with the Faculty Leadership Award. That's a nice short title. Recognizing not only her scholarship and teaching, but her extraordinary institutional citizenship, and I think the presence in this room attests to that as well. She served as department chair for 8 years, bless you, founded the Chemistry Center that now serves hundreds of students every semester, chaired the college chairs, served as associate dean for academic planning, and people still like you, and was one of 4 faculty leaders who guided our academic team through COVID-19 pandemic. Most recently, she has taken on the role of program director for our Worrell House in London. I'm especially looking forward to today's lecture where we get to see Christa as both teacher and scholar in the same room at the same time. So please join me in welcoming Dr. Christa Colyer, who's going to talk about beyond measure chemical analysis in a complex world. And while Christa is up here, I'm going to present her the award.
Thank you. Thank you. Yeah, thank you. Wow. Thank you, Dean Crassus, and thank you to all my friends and family and colleagues and students who are here. Obviously, that's a very humbling introduction, and it's a huge honor to be able to share with you all things that I'm really passionate about, but of course, it's not my own work, it's the work of my students primarily, who are the ones who are toiling away in the lab, coming back day after day to try to reproduce things that don't make sense, but we have to make sense in the end. And so they're the heroes of this over all these years. And of course, additionally, the support, encouragement, and belief of all of you in the same mission that I believe in, which is that we can really, truly help our younger students learn to find their way in the world more than just know a lot of chemistry. So thank you to all of you for being here. My talk today is pretty broad. I want to talk about the world of chemical analysis, because that's where I live most of my time, but think of how it fits into the broader world as a whole. So my talk is called Beyond Measure, Chemical Analysis in a Complex World. And I want to start with a quote from A.E. Hausman, who was a British poet, but also Latin scholar. I don't know if Hubert McNeill Poteat ever read any of Hausmann's work, but it's a very stern first quote, and I was wondering if I should put it up. It's kind of like an admonition. But accuracy is a duty and not a virtue. I think it's such an important starting point, and I hope throughout this talk I can convey that accuracy is not something that we, especially in the world of chemical analysis, should give ourselves a gold star for. It has to be a starting point on which it's our presumption, right? It's our base, it's our foundation, and then we can do great things from there. But I think we can apply that beyond just chemical analysis, and maybe more so now than ever. We really need to think about trustworthiness and accuracy as a starting point. So I thought I would start there, even though it's a little harsh, and I probably would not start at that point in my class. So I'm going to have a 4-part talk. I'm just going to start with measure to begin with, because I think if we can think about what do I mean, at least by measure, we'll all be on the same page as we go through the slides. So measurement, just assigning a numerical value to a property. So length, mass, temperature, using standard units and instruments or appropriate tools to do that. So instead of saying it's hot outside, it's 100 degrees Celsius outside, right? So a measurement, assigning a numerical value. It is hot. It's not 100. That was inaccurate. To be clear. Okay. It comes from the Greek metron, and we measure things all the time, not even thinking about it. For example, many of you probably have a smartwatch on. You check your steps. That's also not very accurate, probably, if we're talking about accuracy, but it makes us feel good. And so we do this. We sort of encounter measurement all the time. So I want you, we're going to start with an activity. Sorry, I'm not going to collect any papers or anything. But I want you to turn and talk to a neighbor, or turn behind if there's no one beside you. 30 seconds only, I promise. And I want you to think of all the ways you've encountered measurements today, just since waking up this morning, whenever you woke up. A measurement you made, or some measurement that you read, whatever it was. 30 seconds. Okay, so call out. What measurements have you made? What measurements have you encountered so far today? Anybody want to shout something out?
Number of students with advising holes.
Number of students. Nice. Dr. Hinsey.
Measured out half a cup of cat food.
Half a cup of cat food. Okay, nice. Others.
9,000 some steps.
All right. Oh, that sounded like a challenge. You know, someone over there is going to say, I've got 10,000. Yeah. Okay, so we encounter them all the time, right? We're familiar with them. We need to think about how we can use them now to solve problems. All right, so where did this begin? Measurement is nothing new, even though we're really familiar now with sophisticated instruments. If we go back to, ancient times, you would say we put a dash of something in, a potion or something like that. But in the Renaissance, I think, was when we started to think beyond just what was in there, and I'm looking at Monique O'Connell, who helped me with this. Paracelsus argued the dose makes the poison. That's not a direct quote, also not accurate, but close enough. The idea of how much, right? So anything could potentially be a poison if you had enough of it. So the amount suddenly started to come into play in our consideration. Through the scientific revolution, so thinking about Galileo and Hooke, thinking on the very big scale of astronomy, the very small scale of microscope, starting to use instruments to extend just beyond what we could see. What we had taken for granted was all that we could see at that point, up until that point. And then the Enlightenment through the Industrial Age to really demand reproducibility based on standards, and we'll talk about what standards are still in play from those days. And then, of course, modern science, where we find ourselves, where my students work every day with really sophisticated tools and statistical analyses, using digital technologies and increasingly now AI technologies, which comes with another talk, which we won't talk about. So, two kinds of measurements. Sorry, the white is hard to see there. Direct measurement versus indirect measurement, if we just put them into two really big buckets. So direct measurement, things that you can see directly or touch directly or use a measuring tool or stick to interpret and assign a number. Or indirect measurement, a quantity that we cannot see directly or measure directly, but by a known relationship with a standard, we can assign a number to it. Archimedes is the sort of classic example of this. He was given the problem to solve whether the king's crown was made of pure gold, but the crown, of course, does not have a standard volume. He couldn't measure it. He sat in a bath, displaced water, and had a eureka moment. Supposedly he yelled eureka, I don't know. The water overflowed the bath, and the volume that overflowed by displacement must have been the volume of his irregularly volumed body. So he acknowledged then that I could measure the crown volume. Comparing it to a known mass, he could find the density, and then he could figure out that the king, in fact, had a crown that was not made of pure gold. Okay, so how do we then assign units to these measures? So this really happened in the sort of scientific revolution period, 1795, thinking about standardizing units globally, and the metric system was defined in 1795 in France. It was fully a decade later before we realized we actually needed some artifacts or some comparators to say that a kilogram in America was the same as a kilogram in France, was the same as a kilogram in Mexico, wherever it happened to be. And so in France, the international prototype kilogram, Le Grand K, was formed. So it's a 1 kilogram platinum iridium cylinder that was housed in bell jars in a cave, in a vault, in just outside of Paris. That was pretty impractical. You couldn't go there every time you needed to measure a kilogram. So countries would standardize against that to provide them with local standards. Again, a physical artifact is not ideal for having to use as a measurement comparator. And so it was very recently that using a natural constant or a constant of nature, Planck's constant, that we use in lots of our calculations, and that could be used now to define the standard kilogram by looking at electrical and magnetic forces in a microscope. So who is responsible for keeping these up? There are agencies all over the world, bodies all over the world. This is like something that is real, like you can get a job doing these kinds of things. And the one that's really responsible for global standards is the BIPM in France, and then the US standards are managed through NIST, or the National Institutes of Standard and Technology. So here's how we think globally on trustworthiness of units and standardization. So we're going to do a little activity here and see how trustworthy our measurements can be. So if you have your camera, and if you're willing, we have a jar of M&Ms, and whoever's closest on this measure can take this home. So there's a reason to pull out your phone. But you don't have to, please, you don't have to. So I just want you to guess the number of M&Ms in this jar. The number of M&Ms in this jar. So I'll give you a minute, Best guess. So this is not really a measurement, right? This is an estimate, but we're going to look at the power of multiple sampling, right? If we have multiple people making a measurement, can that help us to converge towards something that's more accurate than any individual could do? Has everybody got it who wants to get it? Okay, so I'm going to go and take a look at your results. We will take a look at your results. All right, so this might be small. So this is the feedback from the Google poll that you just responded to. So you can see your distribution. I've never done this before. And that is the best bell curve ever. Oh my gosh, that is super lucky. All right, so true count 654. True count 654. Anybody? Okay, come up and collect your M&Ms after the show. Okay, so 654. So we're going to come back to that true count. Think about how we could have counted that. We could have just counted them out one by one, which indeed, thank you, my husband did. He really did. He counted them all. I didn't ask him to do it in triplicate, which is what I would have asked my students. So 654, but you could have imagined just taking a smaller subsample, right? Just taking a portion of the M&Ms, weighing those, comparing them to the whole, and scaling up, and hoping that that's representative. Or a volume and scaling up. Then you run into, are the samples homogeneous? Are they representative? So we start to have problems with those measurements. So here we had 46 responses, and the mean guess was 450. So we'll talk about that. It's not very accurate. But the standard deviation and the standard error help tell us something about the range or the spread or the precision. The smaller the spread, the smaller the range, the more precise the measurement is, or the more reproducible the measurement is. And typically when we have no bias, no systematic error, then the mean, if we have a lot of measurements, will converge to the true value. But the error, and I've got it shown as a percentage error here, is really a representative measure of how far is the true from the mean. That's what we're looking at there. So we undershot it, that's why it's a negative, but nonetheless our distribution is spot on. So that looks nice. Okay, let me go back to our display. Okay. So, just from the measure, we're going to go on from here, but just from this first part, I wanted to highlight, you know, this, what we're talking about here, measurement and accurate measurements are nothing new. It's just trying to move them forward from the rather inaccurate medieval and Renaissance period. And over time, of course, our instruments have allowed more precise measurement. Units have been standardized so we could replicate and trust our measurements. And I had to put this here because every single textbook in chemistry, I'm sure even graduate level textbooks have this picture in them. We use it all the time to distinguish and help differentiate between something that's accurate and precise, which is what our goal is. Accurate, telling us how true something is, how close to the true value. Precise, meaning it's replicable. But often we end up in this category where it looks good. We're fooled into thinking it's really the right answer because it's repeatable, but there's some sort of systematic error that's pushing all of the numbers askew. And that's problematic if we don't identify and control for those. Or we're still, and I have to, no, I'm not gonna say, but Gen Chem, this is where we start, right? Gen Chem lab starts here. It's not accurate or precise, but they're learning the techniques to move across the spectrum, right? And just helping them understand that that's normal. We don't all start out being able to make really good A-level measurements, right? And that's pretty normal. So anyways, measurement has helped us connect from long ago to modern day. So what do I mean by beyond measure when we think about measurement, what we can see? I want to talk about ensuring observational accuracy, even if it's something we can't see. Revealing the unseen, how do we do that if it's not something that we can visually mark on? And I'm going to talk about 3 tools that we use in our lab. There are many, many others. So again, sorry for the slide colors you're seeing here, but differentiating now between qualitative and quantitative observations as we take data in, as we take measurements in, or observations in. Qualitative being descriptive of a quantity, and quantitative, again, putting that number. So remember, we had the M&Ms. I want you to turn to your partner again and come up with a long list in 30 seconds of anything you can qualitatively use to describe it and anything you can quantitatively assign to it. Does everybody know what they're doing? Yeah? 30 seconds. Come up with a list. Qualitative, quantitative. Okay, so we're going to come back. We're going to come back together. We're going to come back together. So tell me about some qualitative observations you've made here. Qualitative observations. Yell it out. Colorful.
Shiny.
Shiny. Shape round. Tasty. Oh, so we're going to come back to that. Tasty. We're going to come back to that. Good. Okay, quantitative. Oh, sorry.
The colors are arranged to form stripes.
Yeah, so a striped pattern. Good. Okay. What about quantitative? What can we say quantitatively? Maybe having to remember back to that spreadsheet you saw. 654. He's a numbers guy.
Mass.
5 colors.
Mass.
Yes, the mass of them, right? Which I can't remember what it is, but they're on there. Yeah, good.
4 calories each.
Oh, 4 calories each. Okay, I'm going to come back to that. Taste and 4 calories each. Nice. Okay. So, very first week of my first year seminar, I teach a first year seminar called Scientists Born or Made. We talk about some of the skills and traits and attitudes of scientists, and observation is where we start because we know that without a strong foundation of reliable, trustworthy observations, then we're going to be doing all the rest of our subsequent measurements and calculations in error, right? If we don't have strong, true observations to begin with. So we start with this book, SS Carey. We start at the beginning of it, and we begin with doing some observational activities. And these are the questions that Carey says we need to ask ourselves. Do I have a sense of what the relevant phenomena are? So you had to know I was asking you about that jar of candy, not a jar of candy you were thinking about from yesterday. Have you corrected for anything that's overlooked? Maybe there's something under the lid that you can't see, right? You've got to worry about that. What's known for sure and what's based on conjecture and assumption? They're tasty. That's based on probably assumption from prior experience, right? Have you considered necessary comparative data? So comparing, different flavors to the side, different types of M&Ms to these, and have your beliefs or expectations influenced your observations? We are human, so we bring that with us. So we're going to now test your observation skills. This is fun, though. This is fun. And we're going to watch this short video. It's 3 minutes, and there's a task assigned to you in the video. So everybody can just personally complete the task, and then we'll report out at the end.
Today we're going to play a game specifically designed to test your spatial awareness. Meet the Brain Games Double Dutch team. What does jumping rope have to do with the brain? Well, Double Dutch requires off-the-chart spatial awareness. Today, these kids are going to help us test yours. For this game, all you have to do is keep track of the number of times that either of the girls in green jumps. You'll count each time one of them lands a jump, like this.
1, 2, 3, 4, 5.
As you can see, these jumpers are pretty quick on their feet, so you're going to have to pay attention to keep up. When the whistle blows, start counting. Ready? Go. So, how many jumps did the green team make? Did you say 38? If so, you agreed with 40% of our test audience. Now, some of you may be onto us, but for those of you who aren't, did you happen to notice anything else going on during the Double Dutch? Maybe a giant chicken strolling right through the middle of the set and doing a funky chicken dance? Now, some of you may have missed that funky chicken, but many of you probably saw it, and that's okay. Because the chicken was just there to distract you too. Here's the real question. What color was the wall behind the Double Dutch game? Here's a hint. It wasn't the same color at the end as it was when they started jumping. The back wall was changing color the entire time, from bright blue to bright red. Nearly everyone misses it. But why? It turns out there's far too much information coming in through the eyes at any given moment for the brain to fully process all of it. As a result, the brain has to act like a spotlight, focusing our attention on some parts of the scene but not others. Now, most of you were probably paying attention to the jumpers, and some of you may have suspected that something strange was going to happen, and so you saw the chicken. But you probably weren't paying attention to the back wall. And what we don't pay attention to, we don't see. And hey, for those of you who managed to catch everything so far, did you notice that we also swapped the rope turners out halfway through? Gotcha.
Right. So I love the statement, what we don't pay attention to, we don't see, right? And that's the human part of it. We bring our biases, we bring our assumptions to all that we're measuring. And we have to check that. That's part of that not virtue of starting at accuracy, right? It's sort of catching ourselves with that.
You never told us how many times they actually do it.
It was 38. It was. That's the actual number. You had to double the 2 greens at the
But
end.
you had to not count the jumps after they got between
Yeah.
the rows, because I counted those.
I know it. Yeah. I'll share the link so you can verify. Collect those numbers. Yeah. All right. So we're going to talk about this idea of, right, what you don't notice, but now we're going to talk about things that are really, really big and therefore can't be noticed necessarily with the naked eye, and we'll talk about really, really small. So Tycho Brahe was the first to make really precise naked eye measurements astronomically, and then followed by Galileo, who used telescopes to be able to make measurements of celestial objects that had never been seen before. We fast forward, it's a long fast forward, to the James Webb Space Telescopes, which have incredible images on their website. Now we can see moon-forming disks around an exoplanet with light that left there 625 years ago. So our snapshot is what happened there 625 years ago. That's how distant it is that the photons took that long to reach our detectors. It's just mind-blowing. We can't imagine we would never have imagined making measurements on that scale. It's phenomenal. And on top of that, we can do spectroscopy, which I'm going to talk about, to determine what are the chemical elements in those clouds. So the other side, or the other extreme, is the small things we cannot see and therefore couldn't know that they exist. So Robert Hooke's Micrographia publication in 1665 documented, and I think was maybe the first use of the word cells, I think, maybe, is that right? And incredible detail looking at fibers, just so beautiful, detailed images and drawings. And then von Leeuwenhoek's single lens microscopes were really the first to reveal microorganisms. So seeing things, and you can imagine, like there's nothing in that water, right? Why am I getting sick from drinking that water? But there is stuff in that water. And it's hard to convince people that there is stuff there when they can't see it with their eyes. So seeing beyond our own eyes. And of course now, fast forward, scanning, tunneling microscopies, electron microscopies, force microscopies, allow us to see truly to the nano level and atomic level, so we can start to resolve images that we just, again, we couldn't even have conceived of. So I love this. I use this slider in my classes because it helps us to get a relative scale. It's one thing to look at powers of 10, 10 to the 18, this giant number of, that's the distance of the exoplanet, or 10 to the negative 12. And we sort of see these numbers, but they might not make sense to us in terms of scale. So this scale bar, I think, and I'll just slide through it quickly, gives us something concrete that we all can recognize, right? A coffee bean, so starting on the 12 millimeter scale, a centimeter scale, and I'm just going to slide through. You'll see all sorts of familiar things. So sesame seed, grain of salt, then we get down to amoeba, paramecium, human egg. Now we're onto the micrometer scale. That's 10 to the minus 6. A micron is about how thick your hair is. So to give you an idea, just keep on going. These things, like, we can't see, right? But they're all there. Yeast, that's familiar. Now we're on the 4 micron scale. Bacteria we start to encounter. I keep on going. Viruses, smaller than that. And there is an endpoint, which I'm going to get to. There's sugar, glucose, amino acids, and carbon. And I wanted to get down here because one of the materials we work with are carbon dots. So 340 picometers. So imagine if I put a period here, a decimal, and I moved this back 12 spots and filled it with zeros. That's a picometer. So it's really, 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 really small. Like we can't sort of physically conceive of something that small. We use numbers in order to explain it, but I think it's hard to really wrestle with the real world picture of that. All right, so how do we see beyond? How do we see these things in our case that are not visible to the eye? We use absorbance spectrophotometry, which measures light, the interaction of light with matter, and it depends on concentration. So this is one of those indirect measures. We want to C, the concentration, so we measure A, the absorbance, right? So that relationship, Beer's law, a known relationship, lets us draw that quantitative measurement, connect the dots, just using light, which goes through a prism or a light selector, passes through our sample, and then is measured at a detector. So the difference of the light in and the light out tells us how much stuff must have been in the cuvette, even in the sample, even though we couldn't see that stuff in the cuvette. So after a sample absorbs light, it's got to do something. The absorption of light promotes its electrons to a higher energy level. Things don't like being in a higher energy level. They have to relax down. Usually they just relax down by bumping into other things or having some sort of vibrational change. But in this case, fluorescence, in some cases, for some molecules, the light will be given off as another color of light or another energy of light. And that's what we use in our lab in order to detect a lot of different chemical species. And so the relationship is very similar. The fluorescence is just some constant, numerical constant, times this phi value. That's called the quantum yields, just how well basically the light is recycled by a substance. But again, you see that direct comparison or proportionality, concentration is proportional to fluorescence. So if I see a higher fluorescence, I must know that there's more concentration or more stuff in my sample. The instrumentation is very similar for those. This is just an absorbance fluorescence diagram. I'm going to go through that because I think I'm going to run out of time. And so here's our carbon dots, and I'm going to tell you about carbon dots next slide. As I have more of them present, so we see the concentration is increasing, the signal, the fluorescence goes up, and we can make a linear calibration curve. So that's how we can see the unseen, in this case, our molecules in the cuvette. So we're going to talk the second tool that we use about carbon dots, carbon nanomaterials. This is like the coolest thing. Carbon can take lots of forms. We know that diamond, right, is a form of carbon, charcoal is a form of carbon, all of these different forms. If you take carbon with some oxygen, hydrogen, carbohydrate sources, and put it in the oven, you probably made carbon dots at home and didn't even know this. You can, and there's some purification, sure, like 3-day dialysis and all that. I know, there's a little more to it. But you can produce carbon dots. We use nitrogen, another element, to dope the carbon dots to give them some different properties in terms of how they absorb and emit. This is not to scale. This is a nanomaterial. So it looks like a beach ball. I realized I probably should have made it look smaller. But nonetheless, here's just plain water. Here's a normal carbon dot sample, and here's our nitrogen doped. And when we hold the UV light behind, you can see the enhanced fluorescence that the carbon dots deliver. So it's just a tool, a lever that we can use by its association with other things in order to be able to see those other things. Those other things themselves, whatever we're interested in, are not lighting up for us. Very few molecules naturally light up. So if we stick them together with our carbon dots, now we have this beacon or probe that allows us to see the unseen. And we use some of the tools we were talking about, transmission electron microscopy, that microscope image we saw earlier, to help us again understand our instruments. So again, instrumentation is used in so many different ways. And then the third tool is a separation tool. So imagine instead of this, you guys were counting M&Ms. Imagine we had this, which is a more realistic sample, right? It's all mixed up, it's a mess, and if I now want to make a measurement of the red M&Ms here, it's going to be a lot harder unless I separate them out first. So a tool that's really common in chemical analysis and allows us to tease apart those things, or the needle in the haystack, is indeed a separation tool called capillary electrophoresis. And so it's a tool that uses a high electric field. There's our voltage that we apply across a length of a capillary, and the velocity or the speed with which the red M&M moves will be different than the speed that the green M&M moves based on this proportionality constant, which is mu, or the mobility. It's sort of how natively well things swim on their own when there's an electric field. So I have a piece of the capillary that we use. I can pass it around. It's made of quartz. It's made of fused silica or glass, but it doesn't break because it's covered with a polymer. It's about 10 micrometers inside diameter, which is, again, close to your hair diameter. So it's hollow. It's like your pipe below your sink in your kitchen, except it's a really, really, really small pipe because we just have molecules going through it, that's all. So you can pass it along. So this ability to swim, as I said, this mu factor, depends on a bunch of different things, but if it boils down and we substitute in a bunch of the normal constants that we use, you can see now that this mobility, how fast something moves in an electric field, depends on Z, the charge. So it's a really highly charged species, 2+, 3+. It's got a lot of electric charge on it. It's going to move faster. And if it's very big, R is the radius. If it's very big, it's an inverse relationship. So if it's very big, it's going to move slower. So we can sort of use those two different measures of an ion to help differentiate and hopefully go from this back to this is the goal. So those are the three tools that we use. I want to tell you now about my lab's broad mission, and then I'm going to talk about some case studies, just some short case studies from our lab. I call our lab analyte agnostic because it's true, we hope in any information that we create, we hope it can be used widely, right? So we try to develop tools or methods that can be used for a whole wide range of different analytes. Research mission statement, this, I'll read it for you and I'll say a little bit about it, is to facilitate the rapid identification and characterization of ligands and fluorescent probes specific to targets that range from the molecular to the microbial for applications to human and environmental health and well-being. So super broad mission, again, that idea of being able to work in different fields and try to be the tools people that can help others solve their problems. I think as teachers, we always think about our learning outcomes or learning objectives. We put them on the head of our syllabus. We make sure students know what they're getting into, what we hope they'll achieve by the end. But we need to think about that for our labs too, right? Or our scholarly projects, just in the same way. So that's what this really represents. As analytical chemists, we focus on these three domains: detection, separation, and selection. So detection, making sure we can see the things of interest; selecting, pulling that needle selectively out of a haystack and not pulling the wrong thing out mistakenly; and then separating them, of course, to facilitate those applications. So here's a range. I went back through some of our publications and just pulled out the names of various analytes. So analytes are the things you're interested in measuring. So this whole thing is a sample, and each of the types of molecule, or M&Ms in this case, are the analytes. So the red analyte might be what I'm after out of this entire sample. And so you can see it's been a wide range. For example, cyanobacteria. These are bacteria in algal blooms, ocean blooms. I've never taken an oceanography course ever, but I got a call, out of the blue from an oceanographer who said, oh, we are having these problems with our satellite images. We think we've got these multiple proteins that are, interfering with our signal. Do you think you could separate them? So I said, yes, of course, right? But it's so cool because you get to learn over all of these years so many other fields that we just never would have had the chance to learn. So in here, pesticides, drugs, proteins that are perhaps, biomarkers, sugars, all sorts of things. It's amazing how many things we get to work on. All right, so one story, and I'll just go through this quickly, and I'm happy to show data slides. I have lots and lots of data slides I can show you anytime in Salem Hall. So this is a story about transferrin, which is an iron-carrying protein, an iron transport protein, the most prevalent iron transport protein in plasma. And this is a picture of it, an X-ray structure of it, or ribbon diagram of it. It has 2 lobes. Each of those can hold an iron ion, each of those. So if it's fully metallated, we call it holo-transparent. If it's fully metallated, it has an iron here and here. If it's totally demetallited, no iron, both of the irons have been stripped, we call it apo. And it could be partially metallated. You could imagine an iron in just this lobe or in just this lobe. So we thought, and it's not, at least not in UV or in visible light, it's not natively fluorescent. We wanted to make it fluorescent, so we thought maybe we could stick it together with our carbon dots, and depending on the structure of the protein, whether or not it has iron in it or it doesn't have iron in it, it will interact differently with our carbon dots, and we'll see a difference in the glowing or the fluorescence of the carbon dots. And so indeed, when we put, if you start at the top, that's just the carbon dot signal alone, and when we put more and more of the demetallited protein, no iron in it, we see virtually no change. And when we put more and more of the metallated protein, we see a quenching or a decrease of the fluorescence that is here proportional to the amount of the protein. So differentiating between those 2 forms based on how they interact with a fluorescent probe. That was one way we could study it. The other was to see, can we separate them? So that's the carbon dot fluorescent probe part, but can we separate them with our capillary electrophoresis technique? Here, oops, well, I'll go to this one now. Here we have just the demetallited protein, Here we have the fully metallated protein. So the demetallited protein, we expect one peak as it moves through that capillary that's moving around. As it moves or migrates through, we expect it to pass the detector, see one peak for a pure compound. For the holo, we see a little second peak, and I'll talk about that in just a minute, but here's the pure metallated protein. And now when we put those together, we have a mixture, and indeed we see and are able to separate when the carbon dots are present, because remember these proteins stick to the carbon dots to a different extent. We see the separation of the demetallited and the metallated, and we have this third peak, which is the same as this one, and that's the partially metallated peak. So one of the lobes has iron or not. So now we're even able to see beyond just the 2 major forms. So I'm going to skip through this in the interest of time. This is using our capillary electrophoresis tool to discover single-stranded lengths of DNA called aptamers, which can be very specific to pull that needle out of a haystack. They can be specifically binding to one target. And the target story that I was going to show you is for vitronectin, which is a protein which is being investigated as a biomarker for cancer. All right, so last but not least, all of this, beyond measure, chemical analysis is happening in a complex world. So I want to talk about how the world has, I would argue, gotten more complex in my almost 40-year arc. What are the big questions today? They're not isolated to our lab. It's not something that we as analysts can solve, obviously. And again, circle back to our need for accuracy and intellectual humility. So you don't need to read these, but I put these up because I went back to see where did this all begin, research, and it began with my mentor, Keith Oldham, at Trent University, which is in about 2 hours north of Toronto. And this was my first ever publication in 1987 as an undergraduate summer research student. And we talk about the transformational impact of experiential learning, and it definitely was so for me. So I thought I would pull that back. It has, to be fair, full disclosure, 2 citations. It has 2 citations, and I had to get it from off-site storage. Okay, so nobody is actually reading it, but that's okay, because I learned a lot doing it. So, yeah, that's a thing. This is my, whoops, let me go back. This is my first publication from Wake Forest. So once my lab was set up, my first 2 graduate students, Missy Moody and Para Viscari, we started our separation journey together there. Yeah, that was many years ago. And this is my most recent work, which is in progress with a collaborator who we have just recently been able to reestablish contact with through going to a conference. And we're working on a new paper work together, which is very exciting, with collaborators in Japan. So, of course, this was all happening. This is a non-systematic historical review of Wake Forest. So like, if your big moment is not here, I don't mean to exclude you. But I wanted to start back where I started, which was 1986, and honestly, I did not know about Wake Forest University, so I'm just coming clean. Like, I did not know about Wake Forest University until I saw the job ad, and then I researched it, and I was super excited, and the rest is history. So back then, when I was at Trent University, the Wake Forest separated from the Baptist State Convention. When I started was the plan for the class of 2000, which was when Wake Forest issued laptop computers to all students for the very first time. And yeah, so I was part of the plan for the class of 2000. Test optional admissions a decade after that, roughly speaking, a decade after that, Wake Downtown. These are all things that have, significantly affected my experience here at Wake Forest, all in positive ways. And then most recently, our new strategic plan, which is leading us forward to our bicentennial, which is like pretty amazing to think of that coming up soon. Of course, this didn't happen in isolation. The science world beyond Wake Forest was doing some stuff over all this time. So when I was in my undergraduate studies was when the Nobel Prize was awarded for the scanning tunneling microscope. You saw some tunneling microscope or some electron microscope images, so that's showing how long it's been around and the time it takes also for things to build. The Human Genome Project in 2003 was largely finished or declared finished to sequence the entire human genome. And it was about 10 years ahead of schedule, one of the rare projects that happens early, because they were not my CE, but capillary electrophoresis was the basis of that sequencing. So I love that connection. And then graphene Nobel Prize, again, you see that we're using carbon dots, but it's another nano form of carbon, so like all of these tie-ins to work. CRISPR, gene editing, we're more familiar with maybe most recently, and last year Science, the journal Science, named the biggest science breakthrough being the surge in renewable energy. So that gives us a little bit of hope that indeed we're going to tackle some of the big problems that we're talking about. Also, this is happening bigger and further beyond us, so going right back to when I was an undergraduate student, Montreal Protocol, that was a global agreement to reduce ozone destroying or damaging chemicals. It really has been remarkably successful. Maybe I think it's by 2040 is the targeted year for the ozone to be completely repaired. It's a win, right? It shows that globally we have the power to tackle these big problems that are beyond any of our individual abilities. The Kyoto Protocol is not as much of a win. We're working hard to make sure we get this. So this was the first global mandate to limit or put targets on greenhouse gas emissions. The most recent update would be the 2025 Paris Accord, really restated in 2015 Paris Accord. This is a big challenge, right? This is a big challenge. iPhone debut, I put this up here because it's such a ubiquitous, tool, treasure, or burden, I'm not sure. You can use lots of adjectives. But it does democratize the ability to have a measuring instrument, right? We can use the camera, we can use the light source. There's all sorts of citizen science projects that utilize the cell phone in a way that gets science out and about and mobile, and that's pretty powerful if we use it the right way. The COVID pandemic, of course, huge impact in so many ways, but especially on science, but also especially as an incredibly powerful story for the importance of fundamental science, right? The vaccine was not discovered overnight, right? So much time and careful, accurate measurement had gone in to make that happen. And then, of course, with the Gen AI explosion, we really have to think about how do we use this moving forward to solve some of our big problems, but ensure the accuracy, and that this is a challenge. So here are the people who did this. An incredible history. Again, very sporadic photos when we manage to remember to do them. And my most recent group this semester, which we all got together on Monday to take. So and really, they're just, it's so fun that we get to do this work with students. Yeah, they keep us smart. So we started here, a sample that, we did our best to try to measure as well as we could. We know in reality this is probably the sample that we're dealing with. And honestly, it's really not that, it's like this. It's just a mess. It's not homogeneous, it's a mess. You don't even like some of the stuff that's in there. And like we still have to deal with it, right? We still have to measure it. So this is the big world. We think about, using the UN sort of framing of the triple planetary crisis in terms of what are the big problems we should all be thinking about. Climate change, biodiversity loss, and pollution. It's like it can just feel paralyzing, right? These are like, I don't even know where to touch these big problems. So we think maybe on a more sort of systems level, things that we can maybe pool together some resources and tackle in the areas of climate, water, food, health, trust data. Again, this is not, we don't own it as chemical analysts, no one of those. We need to all be in all of those boxes together, but it feels a little maybe more like we can bite off some of those bits. And then I really like to go back smaller still, or maybe bigger, I don't know how you define it, but the Sustainability Development Goals, 17 goals, very clear, measurable outcome targets. And I've just starred ones where I think chemical analysis can play a role, and it's pretty substantial. So we just think we can touch somewhere, bring our scholarship somewhere, right? It doesn't feel as daunting as looking at climate change, as looking at biodiversity loss. These are big problems. It is our reality. It is the complex world, but we are just chemical analysts, right? And we still have to feel like we have some power, but we have to also recognize we have limited power. So here are the three key takeaways. What do? We make the unseen seen, we grapple with difference, and we support humble claims. If I left the slide blank, I think every one of you could fill the boxes with your own discipline. Right? I think you all are doing things, no matter what your research area is or your teaching, to make the unseen seen, to grapple with difference and to support humble claims. So we are doing the work, maybe unknowingly together, and I just think it's so important that we know that we are all rowing in the same direction ultimately. So what does chemical analysis do? How do we fit into these boxes in our very small slice? We can use analytical signals to stand in for quantitative measures when direct observation is not possible. We can separate and sense, develop separation and sensing methods to reveal distinctions between and within samples. And we can measure just as a starting point, right? Our accurate measurement just as a starting point to come up with better questions and new directions. And so I'm going to end on a quote just as I started on a quote, circle back to the Hausman quote, and think about this Bronowski quote that says, every judgment in science stands on the edge of error and is personal. Science is a tribute to what we can know, although we are fallible. So I want to thank everyone for